Welcome to the Deep Dive. Today we're digging into some really fascinating new research. It's proposing a fresh strategy against cardiac fibrosis.
That's right.
And for those of you deep in cardiovascular science, fibrosis, this pathological stiffening of heart tissue, it's behind so many heart conditions.
It really is. And we've understood for a long time that this excessive matrix deposition, the stiffness it causes, drives dysfunction. But, actually targeting that stiffness directly in the heart, that's been, well, a huge challenge.
A massive hurdle, which is exactly why this paper we're looking at feels potentially quite significant. Instead of trying to sort of physically soften the stiff tissue.
Which is incredibly difficult.
Right. They asked, what if we stopped the heart cells from sensing the stiffness? Could that be the therapeutic angle?
And they layered on top of that, combining this mechanosensing inhibition with hitting the TGF pathway, which, as you know, is a major player in fibrosis.
A very well-known pathway.
Exactly. The idea being maybe hitting both at once gives you a much stronger anti-fibrotic punch, a synergistic effect.
So our mission in this deep dive is to really break this research down. We want to understand the key findings, look at the experiments they ran, and talk about what this could actually mean for treating cardiac fibrosis down the line.
Sounds good.
Okay, let's jump right in. The core idea really hinges on this positive feedback loop involving stiffness.
Yes. The paper lays this out clearly. It's pretty established now. Whatever kicks it off, fibrosis means too much extracellular matrix gets laid down.
And that makes the heart tissue stiffer and stiffer.
Correct. And this is the crucial part, biologically, the stiffness isn't just an outcome. It's an active signal.
It talks to the cells.
Precisely. It tells the cardiac fibroblasts, the main connective tissue cells, to wake up, essentially, to activate and turn into myofibroblasts.
And those are the cells really driving the fibrosis, right?
Absolutely. They're the ECM factories. They churn out more matrix proteins, which makes the tissue even stiffer, which activates more fibroblasts.
And around it goes.
It's a self-perpetuating cycle, a really damaging pro-fibrotic loop.
And the paper flags key signaling pathways involved in sensing the stiffness. We mentioned TGFβ, but also YAP and TAZ.
Right. YAP and TAZ act like intracellular dials that respond directly to mechanical forces from the matrix outside the cell.
So they convert that physical stiffness signal into biochemical instructions inside the cell.
Basically, yes. Instructions that say, become a myofibroblast, make more matrix. Understanding this loop is fundamental if we want to break it therapeutically.
Okay, so moving from the concept to the bench, they did some really elegant in vitro work using iPSC-derived cardiac fibroblasts to explore this chemomechanical control. How did they set that up?
Well, using human iPSC-derived cardiac fibroblasts is already a great start. It's a relevant human cell model. They first confirmed these cells react to stiffness as expected.
How?
By growing them on hydrogels. These gels were engineered to have specific stiffness levels, some soft like healthy heart tissue, others much stiffer like fibrotic tissue, and of course standard rigid plastic dishes too.
And they added drugs.
Yes, they tested mechanosuppressor drugs and TGF inhibitors in these conditions as well.
And what happened when they put the fibroblasts on the stiffer stuff? Did they change?
Oh, absolutely, just as you'd expect. On the stiffer hydrogels and the plastic, the fibroblasts just spontaneously switched into myofibroblasts. You could see it clearly. They started expressing lots of alpha-smooth muscle actin, ALMA.
The classic myofibroblast marker.
Exactly. That protein is key for their contractile nature. So it clearly showed, yep, stiffness directly drives this activation.
Now, I thought the photoresponsive hydrogel system was particularly neat. Tell us about that.
Yes, that was very clever. They used a special hydrogel where they could change its stiffness using UV light.
On demand.
Pretty much. They could make it softer, quickly, and reversibly. So they could let the cells get activated on a stiff surface first, then soften the environment underneath them.
That lets you ask about reversibility, doesn't it? Can you turn a myofibroblast back?
Precisely. And that's where the key finding emerged. When they combined both things, dynamically softening the matrix and adding a TGF inhibitor drug,
What happened?
they saw a much bigger effect. The myofibroblasts reverted back towards a quiescent, non-activated state much more effectively than with either softening alone or the drug alone.
How could they tell?
Several ways. They had a reporter gene, TGLN, linked to CFP fluorescence. That signal went down. The cells physically changed shape, looked less like myofibroblasts. SMA protein levels dropped significantly, and YAP1, which usually goes into the nucleus and activated cells, moved out.
All signs pointing towards deactivation.
Correct. Strong indicators of myofibroblast deactivation.
And they backed this up with single-cell RNA sequencing.
They did. The cRNA-seq gave them a whole genome view. and it confirmed the story. The combined treatment pushed the cell population profile away from the activated myofibroblast spates and back towards the quiescent fibroblast cluster, called QCF1.
So a clear synergistic effect at the molecular level, too.
Absolutely. It really strengthened the case for this dual-pronged approach, hitting both mechanical sensing and TGF signaling.
Okay, so the in vitro work is compelling. Softening plus TGF inhibition works best. But you can't easily soften heart tissue in vivo. So they needed a drug target, something that mimics the effect of softening.
Exactly. They needed a druggable target that could block the cell's sensing of stiffness.
How did they find one?
Their approach was smart. They did a big meta-analysis, pooling data from multiple single-cell sequencing studies, human hearts, mouse hearts, healthy, diseased. They were looking for genes specifically enriched in the fibroblast populations.
And they landed on SRC, the tyrosine kinase.
Yes, SRC. It's known to be involved in focal adhesions, these structures that connect the cell's internal skeleton to the external matrix. And what was striking was how specifically enriched SRC seemed to be in fibroblasts compared to other known mechanosensors they looked at.
Interesting. Was its expression level higher in diseased hearts?
Not necessarily the baseline amount of SRC transcript in human dilated cardiomyopathy samples they checked, but, and this is key, its activation state was way up.
How did they measure activation?
They looked at phosphorylation at a specific site, tyrosine 416, and in a mouse model of heart failure, the TAC model, this phospho-SRC was substantially increased.
So the protein might be there, but it gets switched on basically when the heart is under stress and getting fibrotic.
That's the idea. The cells sense the stiffening environment and activate SRC signaling.
Makes sense. So SRC looks like a good target. Next step.
Right.
Find a drug to hit it.
Yeah.
That led them to saracatinib, or SAR.
Right. They used computational methods, virtual screening, to look for molecules that might bind to SRC and inhibit it. Saracatinib popped up as a strong candidate from these docking and dynamic simulations.
And they didn't just rely on the computer simulations, did they? There was other evidence pointing towards SAR.
Correct. They used something called the connectivity map, CMAP. It compares the gene expression changes caused by a drug to changes caused by knocking down specific genes.
And what did CMAP predict for saracatinib?
It predicted that SAR would cause gene expression changes similar to knocking down focal adhesion genes and also similar to knocking down YAP1.
Which fits perfectly with the idea that SAR could block SRC-mediated mechanosensing that signals through yapatase.
Exactly. It supported the hypothesis that SAR could pharmacologically mimic some of the effects of matrix softening, at least at the gene expression level.
So theory and computation look good. Did it work in vitro when they tested SAR on the actual cells?
Yes, they went back to the bench, tested SAR alone, and crucially, tested it combined with TGF-β inhibitors SB431542 or pirfenidone on myofibroblasts cultured on rigid plastic.
Yeah, the results?
Very consistent with the earlier softening experiments. The combinations SAR plus a TGF inhibitor significantly reduced the number of SMA-positive myofibroblasts, even when the cells had been activated for a while.
Did SAR alone do anything?
SAR alone did have some effects. It made the cells look morphologically more like quiescent cells on soft surfaces. And the combination treatments were particularly good at kicking YAP1 out of the nucleus and increasing its phosphorylation, meaning less activity. And importantly,
Go on.
the combined treatments also significantly reduced the amount of another key protein, MRTFA, in the nucleus, just like they saw with physical softening plus TGF inhibition.
MRTFA keeps coming up as important in these combination effects.
It does, and they did co-immunoprecipitation studies pulling down MRTFA and seeing what proteins came with it. The data suggested the combination treatments, both SAR plus TGFα and soft matrix plus TGFA, reduced the interaction of MRTFA with other nuclear proteins, including one called SORBS2.
Interesting. We'll come back to that mechanism. But first, did this dual drug strategy work in more complex systems, like engineered tissues or actual animals?
That was the crucial next step. First, they tested it in engineered heart tissues, EHTs. They induced fibrosis and stiffening in these mini tissues. Encouraging results. The combination of SAR plus a TGF inhibitor helped restore some of the EHT's contractile function. It reduced the tissue compaction, a sign of less fibrosis, and actually decreased the measured tissue stiffness.
What about the single drugs?
Much more modest effects, mirroring the cell culture results. The combo was clearly better.
Okay, EHTs are one thing. What about in vivo, in mice?
They used two standard mouse models of heart disease. Myocardial infarction, MI, basically a heart attack model, and the transverse aortic constriction, TAC model, which mimics pressure overload like in hypertension.
And did the combo therapy work in both?
Here's where it got really interesting. No, it didn't.
Right.
In the acute MI model, which has a lot of inflammation and cell death right after the injury, the combination therapy didn't really provide much benefit, didn't matter much when they started the treatment.
Why do you think that is?
The paper suggests that maybe in these very inflammatory conditions, this particular strategy just isn't as effective. Other pathways might dominate.
But what about the TAC model? That's often considered more relevant for chronic fibrosis due to pressure overload.
Exactly. And in the TAC model, the results were much more positive.
Good. What do they say?
The combination of SAR and pirfenidone significantly reduced both the interstitial fibrosis between the muscle cells and the perivascular fibrosis around the blood vessels.
Did it help heart function?
Yes, it partially rescued the drop in systolic function caused by the TAC procedure. And importantly, this happened even when they started the treatment weeks after the pressure overload began, suggesting it could work on established fibrosis, not just prevent it.
That's potentially very relevant clinically.
It is. Though, again, the single drugs alone had pretty limited effects. And one caveat, the benefits seem to wear off when they stop the drugs. It might require chronic treatment.
Okay, and they dug deeper into the why using single nucleus RNA sequencing on these TAC hearts.
Yes, snRNA-seq. This let them see gene expression changes within specific cell types in the treated hearts.
What did that reveal?
It pinpointed a specific subcluster of cells that looked like myofibroblasts and expressed a lot of fibrosis genes. This subcluster got much bigger in the untreated TAC hearts.
And with the treatment?
With the SAR plus pirfenidone combination, this profibrotic subcluster shrank the most dramatically.
So the drug combo is reducing that key pathogenic cell population.
Seems like it. And this reduction correlated with lower expression of matrix genes and also targets of all the pathways we've been discussing. TGFS, S-AMAT, MRTF-SRF, and YIPTZ.
All suppressed by the combo therapy.
Most effectively by the combo therapy. The single drugs had, again, only modest effects on these gene programs in vivo.
It really reinforces the synergy idea at the molecular level within the whole organ.
It does.
Okay, let's circle back to the mechanism of that synergy. You mentioned MRTFA and this protein SORBS2. How do they think the combination therapy works through them?
So the thinking is that the combo therapy really puts the brakes on the MRTF-SRF pathway, which is crucial for driving the myofibroblast state.
And why does it take the combination to do that effectively?
Well, their data suggested that either TGF signaling or just being on a stiff matrix was enough on its own to get MRTFA into the nucleus, where it does its work. There seems to be some redundancy upstream.
So blocking just one input isn't enough to shut it down.
Apparently not completely, but the really novel finding was this interaction they uncovered between MRTFA and SORBS2 happening inside the nucleus of the myofibroblasts.
SORBS2 is a cytoskeletal protein, isn't it?
Right.
What's it doing in the nucleus interacting with a transcription factor?
That's the intriguing part. They showed this interaction happens, and importantly, the combination therapy seemed to disrupt it.
And what's the consequence of disrupting that interaction?
It seemed to make MRTFA more mobile within the nucleus and ultimately led to it being exported out of the nucleus and into the cytoplasm.
Make it away from the genes it's supposed to be activating.
Precisely, reducing its ability to drive that profibrotic gene program. And they did another experiment where they knocked down SORBS2 using genetic tools. Knocking down SORBS2 also led to more MRTFA being found in the cytoplasm and made the MRTFA still in the nucleus more mobile. It mimicked aspects of the combo drug treatment.
So the model is SRBS2 somehow helps anchor or retain MRTFA in the nucleus, keeping it active, and the dual therapy messes up that retention mechanism.
That seems to be the working hypothesis, yes. By hitting the upstream signals, TGFA and mechanosensing via SRC, and potentially disrupting this downstream MRTF-SRABS2 interaction, the combo therapy achieves a more complete shutdown of the fibrotic machinery.
Very interesting mechanism. Now, thinking about potential clinical use, what about side effects or selectivity? Did the treatment seem to harm other heart cells?
That's always a crucial question. Based on their observations, the combination treatment didn't seem to cause obvious adverse effects on the overall heart structure or function in the mice.
And why might that be?
They suggest it might be because SRC expression and activity are relatively low in the other main heart cell types,
Right.
like the cardiomyocytes and endothelial cells, compared to the fibroblasts, especially in the fibrotic state.
So it's hitting the fibroblasts, the stromal cells, more specifically.
That's the hope. a degree of stromal targeting selectivity, which could minimize unwanted effects on the muscle cells themselves.
And this leads to the idea that maybe this approach isn't just for the heart.
Right. The authors propose that this strategy targeting mechanosensors enriched in stromal cells, combined with blocking key profibrotic signals like TGFβs, might be applicable to fibrosis in other organs, too.
Like the liver, lungs, kidneys.
Potentially. Fibrosis shares common mechanisms across tissues, and the role of mechanobiology is increasingly recognized everywhere. So this could be a broader conceptual framework.
Okay, so let's try to wrap this up. To synthesize this deep dive, the study makes a really strong case for this dual mechanotherapy idea for cardiac fibrosis. Inhibit SRC to block stiffness sensing and inhibit TGF signaling.
And they showed synergistic effects across the board in cells, in engineered tissues, and importantly, in vivo in the TAC mouse model, where it reduced fibrosis and helped heart function.
But we need to remember the limitations. It didn't work well in the acute MI model.
Right, highlighting that the context, especially inflammation, really matters. And there's still more to learn about that MRTF-SORBS2 interaction. Plus, the in vivo benefits required ongoing treatment in the study. Long-term effects and safety need more work.
And we always need better tools to really track fibroblast behavior in vivo.
Absolutely. Characterizing fibroblast dynamics and heterogeneity is still a major challenge.
So this research definitely opens up exciting avenues. It makes you wonder, doesn't it?
Yeah.
Given these findings, how could we refine this specific dual approach? Or could we find other, maybe even better, stromal mechanosensors to target?
Yeah. Are there other key players in that fibroblast mechanosensing network we could go after? Perhaps ones that offer even greater specificity or potency?
And what are the really critical next hurdles to even think about translating something like this SAR plus an anti-TGF drug towards actual clinical trials for patients with heart failure and fibrosis? Lots to think about.
Definitely. It's promising work, but still early days in terms of clinical reality. Fascinating science, though.
Absolutely. Well, thanks for joining us for this deep dive.